Okay, so for today we are going to cover subtopic 6.3, which is pollination and fertilization. Okay, pollination and fertilization. The process in which pollen grains are transferred from the anther to the stigma is known as pollination. This process is assisted by pollinating agents such as insects, mammals, birds, water, or wind. The presence of pollen grains on the stigma triggers the process of fertilization.
So here is a picture of the formation of pollen tube and male gametes. So I'm going to explain a little more details about it. First, the wall of anther from matured pollen will dry, shrink, and split. Pollen grains in the pollen sac are released. The released pollen grains are transferred to the stigma of the same flower or different flowers by a pollinating agent. So we get to the picture here. This is anther, right? So the wall of anther will mature, dry, shrink, and split, and will release the pollen grains. The pollen grains are transferred to the stigma and also transferred to the different flowers by pollinating agent. After that, pollen grains that have been transferred to the stigma will germinate and form a pollen tube. So this is the structure of pollen tube. This is pollen grain and the structure of the pollen tube. The pollen tube grows towards the ovule through the style. Then the generative nucleus will move along the pollen tube towards the ovule. Okay, let's see here the mature pollen. The mature pollen consists generative nucleus and also tube nucleus. So, first of all, the tube nucleus will lead the formation of pollen tube. After that, at the same time, the generative nucleus will divide into two male gametes, so there is no generative nucleus. Next, what will happen during the formation of pollen tube is the end of pollen tube will secrete an enzyme to digest the tissues of the style. That's why the pollen tube will form by using the enzymes. Okay, this is the picture where the pollen tube enters the micropyle when it reaches the embryo sac. And then the tube nucleus will degenerate. So look at the picture. There is no tube nucleus, only two male gametes because the tube nucleus will degenerate and both male gametes enter the embryo sac to carry out fertilization. So this is how pollen tube developed before the fertilization process.
So this is some example of differences between self-pollination and cross-pollination. First, transfer pollen grains from the anther to the stigma of the same flower. This process can take place in either in the same flower or another flower of the same plant. It occurs in the flowers which are genetically same. Occurs in perfect flowers, causes homozygous condition in progenies, increases genetic uniformity and decreases genetic variation. So this is for self-pollination. Okay, next is cross-pollination. Transfer pollen grains from the anther to the stigma of the different flower. This process can take place between two flowers on different plants. It occurs between flowers which are genetically different. Occurs both perfect or imperfect flowers. Causes heterozygous condition in progenies. Decreases genetic uniformity and increases genetic variation. So this is for cross-pollination.
Okay, double fertilization. What is double fertilization? We call it double fertilization because it involves two times fertilization, which are first male gamete fertilizes the egg cell to form diploid zygote. Okay, look at the picture. This is the first male gamete which fertilizes with egg cell to form zygote. And the second male gamete here will fuse with polar nuclei. This is polar nuclei to form triploid endosperm tissue. Let's go detail for the double fertilization. Okay, first, when it reaches the embryo sac, the pollen tube will penetrate the ovule through the micropyle. So, this is the pollen tube reach the ovule and penetrate the ovule through the micropyle. After that, the tube nucleus will degenerate and move male gametes into the embryo sac. One of the male gametes fertilizes the egg cell and produces a diploid zygote. In the second male gamete fuses with the two polar nuclei to form a triploid endosperm nucleus. In summary, first fertilization is first male gamete fertilizes with itself to form diploid zygote, and the second male gamete fuses with the two polar nuclei to form triploid endosperm nucleus.
So last but not least, what is the importance of double fertilization for the survival of flowering plants? First, produces diploid zygote to ensure the species genetic content is maintained. Second is to ensure development of endosperm tissue only in the fertilized ovule. Third, produces triploid endosperm nuclei to store food for seed germination. Lastly, create variation through meiosis and random fertilization of gametes.